This title to upset some people in the room. I imagine you all are saying, no, absolutely not. Aging is not contagious. However, I think millions of people around the world would beg to differ. Long COVID has become something that is actually being reported now. It's millions of people just in the United States. And you can see very clear evidence of aging in these patients. You see frailty. You see many of the other hallmarks of aging, which I'll be going through today. So this conversation is really going to be reviewing the known pathogenic drivers of degenerative aging going back decades. And so this QR code here, I'm going to be hitting on many dozens of different studies. And so you'll want to look at those studies later. The QR code will allow you to link to those, and then you'll be able to download those studies. So the technology that we're working on is a deep tech breakthrough with an AI drug discovery engine that may stabilize the microbiome and extend health span up to about 120 years. And so we're relying on others to get past the 120 out to hundreds of years. But we believe just this technology alone that we're going to be talking about can extend health span out to 120 years. Now, I want to start with gratitude and acknowledgments for many of these collaborators and partnerships with the Department of Energy, U.S. Department of Defense, Stanford University, NYU, University of Wisconsin, Texas A&M, and many others that are not on here. The NIH has provided many millions of dollars to support this. And so we're going to be talking about aging and pathogenesis, right? So the contagious aspects of aging. The traditional view is that aging is a multifactorial process influenced by genetic and environmental factors that may lead to an increase in vulnerability to pathogens. And so that's essentially what we've been talking about at this conference up until about probably 2022 or so. The emerging view is that aging and the microbiome are intimately intertwined. The data show pathogenic microbiome disorder, right, which can be brought about by antibiotics. It can be brought about by catching COVID. Many different aspects of how the microbiome can be disordered. This is called dysbiosis. So this accelerates aging and may be a key driver of aging. So more acknowledgments here. The Hallmarks of Aging, An Expanding Universe was published in 2023, and it's impacted all of us. It's a beautiful, elegant study. I'd like to thank these authors, especially if they're here in the room today. I'd love to talk with you at some point. I have removed the labels here. So they have traditionally been called integrative, primary, and antagonistic. So I've removed those labels to remove the prejudice associated with those labels. And so we're just going to look at the hallmarks of aging here as they stand. We're going to start with dysbiosis here, and we're going to show, okay, in these studies here that you may want to download, what these studies show is the causative effect of dysbiosis causing chronic inflammation, right? The next one here, dysbiosis causes altered cellular communication. Dysbiosis causes stem cell exhaustion, especially within the gut. Very well studied in the gut, and also in many other organs that are directly connected to the downstream precursors for the various different hormones and other areas that come from the short-chain fatty acids produced by the microbiome in the gut. Dysbiosis also causes cellular senescence. So you've heard from some of the prior speakers in this section on that. Disbiosis causes mitochondrial dysfunction. So mitochondrial dysfunction is very tightly integrated to causing chronic fatigue syndrome in long COVID, for instance, right? So this is not just a bacterial cause, it is also a viral and fungal cause. Right? And so this polymicrobial ecosystem that we have, and we think of primarily just in the gastrointestinal system, is also in your nasal cavity. It's also in your brain. It's also in all of the organs of your body. In fact, every organ in your body relies on its microbiome to function appropriately. So dysbiosis causes deregulated nutrient sensing. Now, of course, this is prevalent in the gastrointestinal system where all of our nutrients come from, right? But it is also a problem in many of the organs, right? So a good example would be like B12 is a vitamin that is produced by the microbiome in the gut. But many of the aspects of B12 are also regulated by the microbiome in the rest of the body. So dysbiosis also causes genomic instability. Now, this is probably the most controversial among the traditional group. So this is very well studied. It's studied in riboflavin studies, the gut microbiome imbalance and base excision repair dynamics in colon cancer. That's a really good one there. And then genomic instability and epigenetic changes during aging. That's also a great study. Dysbiosis causes telomere attrition. So telomere is probably within the sort of layman's audience, telomere attrition is probably the most well-known. It just sounds great, telomere, right? So it's very provable and also has been proven in many dozens of studies here that you can link to, even in children, that telomere attrition is accelerated due to dysbiosis. Dysbiosis causes epigenetic alterations. And so we saw some of that evidence just now in the past talk, but there's many more. There's dozens of studies on this as well. Dysbiosis causes loss of proteostasis. In fact, many of the proteins that regulate homeostasis in the body and the short-chain fatty acids that trigger the genetic expression of those proteins come from the microbiome. Dysbiosis causes disabled macroautophagy. And so this was the one when I was going through these studies, I thought, does it really do that? And so I really dug into that and got rid of several studies that I thought, like, it's just not clear. These are three that I thought are very clear evidence of causative relationships where disabled macroautophagy is certainly caused by dysbiosis. So let's go for it. Strange word, dysbiosis. It sounds like just a disruption of the biological systems, right? Really what it is the disordered microbiome. Now, why is this pervasive and why is it a horrible problem and why do we not really want to talk about it, right? Because we all have a human genome. But how do we control this just chaotic thing that's inside of us, right? Are we the bus or are we the bus driver? Or are we the passenger on the bus? Like, there are more cells within the microbiome than there are human cells in the human body. And so this makes our job very complex, right? Well, we're moving into an age of AI and quantum where we're going to be able to encapsulate this. And then also we're moving into an age of drug discovery, which is what we're here to do, right? Which will help regulate the microbiome. And so that we'll have sort of normative data across the human population where we're reducing the pathogenic influence within the microbiome, and we will have a more commensal microbiome that we can deal with further into the future. Essentially what we're talking about is the end of the age of diseasing. And that's what our company, Maxwell Biosciences, is designed to do. So in viruses and bacteria, what you have is that you've got a big problem here where it takes years and years and years to create a drug. And then in bacteria, you have this ticking time bomb where we know that very soon, in the near future, probably within the next 5 years, most bacterial infections will be antibiotic resistant. In India, which is essentially 5 years ahead of us on that negative curve, right? Most infections are antibiotic resistant, total drug resistant. Just outside of Australia, they have total drug resistant tuberculosis, which is airborne bacteria that there is no drug that can cure and you will die from it. So this is a nightmare, right? Fungi, there are very few drugs that can treat fungal infections because fungi is on the same branch of the tree of life. So we're in the same kingdom as fungi. We're not in the same life kingdom as bacteria, right? And so how do you have a drug that can attack multiple kingdoms within the tree of life and still be selective against human cells? So that's what we're headed towards with our company, with this deep tech breakthrough. So the key point here, without going into too much detail, is essentially that dysbiosis is a driver of muscle frailty, immune senescence, and cognitive decline, which I thought was quite interesting. The X Prize just talked about those are the 3 targets, right? And so this is something I think everybody should be paying attention to, is it that you are going to say that your one therapeutic is going to solve all of this if you're not paying attention to dysbiosis? The consequences of the aging microbiome are you have increased gut permeability. And so essentially what happens here is that you actually are infecting yourself with your own commensal microbiome. So, in the Garden of Eden, the original sin, right, is that we are cursed with this thing inside of us, right, that can infect us. We're carrying around our own pathogenic microbiome potentially, right? And so this is something that we need to solve. It causes systemic inflammation and altered metabolism. So, how does the human body defend against this? There is a natural peptide inside of the human body called LL37 in the innate immune system that has an extremely potent and very broad spectrum antimicrobial properties. It is very effective against fungus, and yet is selective against human cells. It doesn't attack human cells. It is effective against all viruses, all envelope viruses that we know of. It is effective against all pathogenic bacteria, but does not affect the commensal microbiome. And so wouldn't that make an excellent drug? So what's even more interesting to us, gerontologists and folks looking for primary drivers of aging and pharmaceuticals that can affect them, is that it repairs damaged cells and damaged tissues. It is anti-inflammatory. And you cannot have apoptosis, which is the replacement of aged cells, without this peptide. It is the key. It starts the apoptosis process, LL37. So if LL37 is so amazing, why do we need to develop new therapeutics to slow aging if natural LL37 inside of your body works so well? Well, the answer is this is an undruggable peptide. It is so unstable that it disappears very, very quickly, within 6 minutes in some cases, up to around 4 hours or so in other species and in other metabolic states. So LL37 is highly unstable inside of the body because of this carbon chain here, this carbon bond. The side chains are attached at the carbon, and that's where the enzymes come in and snip it. So the carbon bond is definitely the problem, right? So if the carbon bond is the problem, DARPA came in, and NIH and the Department of Energy, and they said, is it possible to create a synthetic immune system? Can we make this more robust? Can we make it so that you don't have to have a cold chain, you don't have to have refrigeration? Is this something that we could give to our troops? Is it something that we could end health disparity in Pakistan and India and Africa? Can we make this very cheaply? Can we not require refrigeration? And it turns out that this is possible. If you attach the side chains at the nitrogen, and this is the big deep tech breakthrough that I was talking about. Any peptide in the body, any peptide that is identified from the microbiome, as we just heard, this can be copied and mimicked accurately, including chirality, with this nitrogen chain. It's called peptoid technology. If we want to look at, we have patents on it, and an AI drug discovery technique. So this is the Clarimer platform. It's this deep tech breakthrough powered by AI that allows us to create anti-aging drugs, virucidal, fungicidal, bactericidal drugs, as well as copy human growth hormone stimulating peptide and many other different types of peptides which we're working on. So there's an AI-guided drug discovery platform behind this that is very interesting. This is what I started on 8 years ago, and I am a data scientist working in AI. I've been working in AI before it was called AI. So let's go into some of the data that was produced by our partners. So what you see here in the, first of all, this is an Ecoli. You have the untreated control down here at the bottom. This is what a regular Ecoli looks like using soft X-ray tomography. In this top image here, you see that the internal ribosomes and the functioning metabolism and essentially the heart and organs and all of the things that make an Ecoli work, it looks like a tin can that has been squished. And that's what our drug does, right? So it essentially halts the metabolism of the bacteria. It eats in whole in the membrane here. But what's interesting is that all the insides, the nucleic acids and everything, are not able to escape because it binds them and aggregates them. And so the nucleic acids are not able to escape outside of the bacteria to inform the other bacteria to create resistance. And so what we see is zero resistance from bacteria to this drug at all. And so you can see over here that this drug is not the fastest at doing that, but it is by far the safest. So viruses have the same vulnerability that your apoptotic senescent cells also have. And so what you see here is in the host cell here, either an infected host cell or a aging cell, they produce this phosphatidylserine marker, which is the apoptosis eat me signal when you're looking at studies based on apoptosis. And so these little black dots here are symbolic of that. And you can see that the viral capsid buds from the host cell membrane and then carries that membrane with it with the phosphatidylserine embedded in the membrane. And that's what your immune system, LL37, and our drugs target. And so we blow up that membrane. We rip it apart. That's what the drug does. And you can see it in these images here. They are in greater detail. Up at the top, you have herpes simplex virus 1, which 75% of us studies show are infected with, and SARS-CoV-2, which we're probably all infected with. And you can see partially disrupted membranes and then fully disrupted there. That viral study there, along with one where we completely suppressed Ebola, sponsored by the NIH, led to multiple U.S. Army contracts. Each one of these are worth over $1 billion. So multiple contracts worth over $1 billion. There's a lot of money, a lot of interest in this space right now. This is one drug for many bugs. So MXB22510 is effective against all tested pathogenic bacteria, including the escape bacteria, all tested pathogenic fungi, many different types of viruses, not all of them, and then we thought almost, most importantly, no adverse effect on the commensal microbiome, which is required for that longevity effect that we're looking for, right? So this is how we compare to existing antibiotics and antifungals. We are far safer and far more broad spectrum efficacy than any of them. We show no resistance. I talked about this before compared to gentamicin. Here you can see fourfold change in resistance across the top there, and then no resistance whatsoever after 30 days of informing the bacteria with our drug, low doses of the drug. So zero resistance whatsoever. And then we also protected against aspergillus. We've tested against many different fungi. It's safe and effective. They're well tolerated with no neuron toxicity in animal models. We've tested in rats and mice. And zero inflammation in animal models as well. Here's the controls over here showing that the test was effective, and you can see absolutely zero inflammation across multiple drug candidates here across the bottom. So this is probably our biggest accomplishment over the past 12 months since the last ARD meeting, is we now have an ongoing partnership with the government of India, the Indian Council of Medical Research, which is their equivalent of the FDA. And they have sponsored the company. They are funding this study. They allocated 24 rhesus macaque monkeys here out of a total of 60 that they have throughout the entire year. So this is almost half of the whole country of India's allocation of non-human primates for this study. You can see the priority that they gave it, right? And so what we do here is we're collecting the baseline microbiome in both of these groups. In group 1, we are infecting 12 non-human primates, rhesus macaques here. We're treating 10 of them with 2 as a placebo. They are infected with multi-drug resistant MRSA, so Staphylococcus aureus. This shuts down hospitals and industries. And then in this, the second group here, we're infecting them with drug-resistant Candida albicans fungi. So this study has already begun. Around midpoint next month, we showed up our preliminary results. And then by the end of the year, you should see the beginning of our published studies on this in high-impact journals. There'll be 6 or 7 studies that come out of this. It's very interesting. It's a first in many different ways, because this one molecule will be essentially published as a therapeutic for treating multiple kingdoms of life. It's the first time in the world that's ever happened. This is how we're going to bring it into humans, is a nasal spray. And so what's really interesting about that is we're delivering it directly on target. We don't have to go through the bloodstream to get it directly to the microbiome in the nasal cavity and in the brain. So 30% of the molecules that leave the nasal spray go into the brain and target the olfactory nerve pathway, the trigeminal nerve pathway, and then also when you swallow, they go down into the through the mucus down into the microbiome in the gut. And so this is what the FDA is looking for here in this study. We're targeting healthy aging. We're working with the U.S. federal government, the Indian federal government, and multiple other governments on biodefense, both antiviral, antifungal, antibacterial, replacing the entire antibiotic industry here. And our key clinical trial is moving forward into chronic rhinosinusitis, which is about a $6 billion indication. Post-viral syndrome like long COVID, infectious dementia, and Epstein-Barr, and brain fungal indications here. So the highlights here are that pathogens are a key driver of all of the hallmarks of aging. We have a single drug candidate going to FDA human trials within the next 12 months. We have over 47 million in government-sponsored research, not including the non-human primate study that just kicked off, and we have 23 million in venture stage investment. We've partnered with the U.S. Department of Defense. We have multiple signed U.S. Army contracts. We're about to sign with the U.S. Navy as well. We have several additional in progress. There's no development of antimicrobial resistance, no adverse effect on the healthy gut microbiome, and no need for refrigeration or cold chain logistics, which really helps with militaries and with ending health disparity throughout the world and replacing antibiotics, which can be expensive in that regard. We're heading into a $6 billion annual revenue opportunity on our first indication, and inside of the United States, they have a qualified infectious disease product that extends the exclusivity of your patents and your commercialization stage by 12 years. I should say to 12 years. And we're looking for partnerships and collaborations to support our FDA trials and commercialization, manufacturing, and distribution, and of course, investment. And I don't know how much time I've got left, Morten, but I'm open to...
We can take a question or two.
Okay.
Thank you so much, Scotch. Really great, inspiring talk. Do we have, does anybody have any questions for Scotch?
It's very exciting, especially as an antimicrobial. I find it very striking, the effects. What I'm curious about is, do we know anything about how it can target specifically pathogens and not other gut microbes?
Yes. So in apoptosis, phosphatidylserine is required for an apoptotic bleb to merge with a cell that's interested in the contents of the apoptotic bleb. And so in apoptosis, you want to, it's essentially recycle and reuse, right? And so the surrounding cells and tissues are trying to accept some of the proteins and such, right? And so viruses, bacteria, and fungi, they also express phosphatidylserine to trick these cells into thinking that they're an apoptotic bleb. And so it's required, it's sort of burglary tools, right? It's a legitimate key, but it's also burglary tools. And so your immune system, your innate immune system targets phosphatidylserine to get rid of those pathogens. Non-pathogenic microbiome do not need to invade a cell, therefore they don't express phosphatidylserine on the external membrane.
Very cool. Thank you so much, Scott. Really a fantastic talk. We are already almost 10 minutes over, so we will say thank you for now.
Thank you.
Give him a warm round of applause.